Okay, welcome back everyone. Is everyone occupied? Anybody still to be waited? No. Okay, then right. Now I can see all the participants here, and I wanted to know the feedback for the first part of the presentation today that we had. You can move like that. What I am going to do, I wanted from you, and you all can listen to every one of you, right? Right? Yeah. Okay, you can unmute just for the time being. And can you tell what techniques that we have discussed in the first session? From the beginning.
You can pick a student and ask.
Nima Sara Ben. First technique.
So the first technique studied was agarose gel electrophoresis, which is used to separate a DNA.
Okay, good. Then Meghna yes.
Yes, sir. The SDS, the protein separation technique, SDS page.
Are you sure? You are skipping, I think. Sneha?
So pulsed field gel electrophoresis for bulk DNA molecules.
Right. Ranjita?
The next technique was SDS, that is the denatured protein gel electrophoresis.
No, you are still skipping. Uma? No, you have to unmute. Unmute. Uma, you have to unmute.
Native phase electrophoresis.
Yes. What are those?
Non-denatured protein, separation of non-denatured protein.
Right. In that, we discussed two of the techniques under native phase electrophoresis.
Yes, sir.
What are the techniques under native?
Activity gels and zymographs.
Right. Then Abhijit. Next, what technique we discussed?
Sir, SDS page electrophoresis.
Yes. So what is the intention? Why you want to do SDS page? When you will do?
Sir, for bulk.
Bulk.
Yeah, bulk DNA.
SDS page. What for you want to do SDS page? When you want to do? Shilpa Soni.
Sir, SDS is for the separation of DNA because DNA is having higher axial ratio than protein.
I hope like when I told in the beginning, you have to maintain the notepad and write the flow and then what are the points at least. Okay. Ishani?
Yes, sir. SDS is for protein separation, sir. First denaturing the protein, then its separation.
Right. So Shilpa and Abhijit, you have to just put some more efforts to understand, okay?
Okay, sir. Okay.
Yeah, right.
Right.
So Shilpa was telling DNA with polyacrylamide, no, but you can't use DNA separation by polyacrylamide. You can use only that is for sequencing. Polyacrylamide will form very small pore size, and that is the technique we used only when you want to sequence. That is polyacrylamide, and it is non-gradient gel. Already we discussed all these things, and I presume I wish that you should know. Okay, so a little bit more efforts. I think you may be getting the slides and go through again. Then next, Deepti. After SDS page.
So polyacrylamide gel polymerization, we have discussed this technique.
Right, SDS page is the technique where we discuss polyacrylamide gel polymerization. Not only that, I told two parts. First part was sample preparation in SDS page, if you recollect. Second part we discussed was about the
Beta mercaptoethanol.
that is all sample preparation, and then polymerization we discussed, right? How the gel polymerization, what is the principle of it, you know now. Then after the SDS page, what technique?
So tube gel electrophoresis.
Right, very good. So that was isoelectric focusing. Then Nilesh Kulkarni.
Field gel electrophoresis, sir.
That's what you have to follow the flow. That's what I said. I said pulse field after agarose gel electrophoresis. The intention was agarose gel electrophoresis and pulsed field, both of these technique used for DNA. Pulsed field is for the bulk DNA for separation. But tube gel, after that, I didn't give the explanation for pulsed field. But what is the technique?
Based on the pH, sir.
No, isoelectric focusing, okay, Dipti has told already. After isoelectric focusing, what was the technique we discussed?
Human electrophoresis, sir.
Okay. Then, after Nilesh, Elizabeth, what technique?
Isoelectric focusing.
Electrophoresis, right? Isoelectric.
After electrophoresis, we discuss about the IEF.
See, isoelectric focusing or tube gel electrophoresis is the same. We had discussion about isoelectric focusing, and then Nilesh has told that immunoelectrophoresis.
Rocket immunoelectrophoresis.
Right, rocket immunoelectrophoresis and
blue native gel electrophoresis.
Immunoelectrophoresis and rocket immunoelectrophoresis are immunoelectrophoresis techniques that we have discussed. And Monisha?
Yes, sir.
What next technique we discussed after immunoelectrophoresis techniques?
So rocket gel electrophoresis.
Which one?
Rocket electrophoresis.
Yeah, afterwards. Akash?
Sir, after immunoelectrophoresis, we were discussing about blue native gel electrophoresis. It is used to separate multi-protein complexes.
Yes. To analyze the monomers in a multi-complex protein. For that reason, blue native gel electrophoresis. We discussed. Okay. Ajas Khan, right? After blue native, what was the technique we discussed?
Rocket electrophoresis.
No, that is rocket is same under immunoelectrophoresis. We discussed immunoelectrophoresis and then rocket electrophoresis. Both are immunoelectrophoresis techniques. After this, we discussed about what? After immunoelectrophoresis, blue native, we told, right? Blue native. After blue native,
Diagonal gel electrophoresis.
Diagonal gel electrophoresis we had discussed. So all those are a major category of electrophoresis techniques, but there are so many other techniques that even you can get the information, like paper electrophoresis technique also was there. That can also be used. And we didn't discuss that because what we have discussed today, those are the having applications in the industry. Okay, so those are some of the techniques we discussed, and all the basic information I had shared with you, and you should improvise your knowledge on electrophoresis, go through the contents and literature and get the further information. So that was just to wake you up if you are not still awakened after gap also. So next technique we are going to discuss, that is capillary electrophoresis or capillary gel electrophoresis. As I mentioned, capillary electrophoresis technique is the third generation technique, which is highly advantageous. Mainly it is a kind of high throughput technique. So wherever the word high throughput means large amount of data within small time. All corporate world works with the time is very important. Within small time, you should be in a position to get large amount of data. So that is the main principle of high throughput. So capillary electrophoresis technique is one of the high throughput. So many number of samples you can run within small time. So this technique was pioneered by Stellan, and in 1928 around his 1960s this technique came, and as we already discussed in the illustration, capillary electrophoresis, the capillaries which you know as you had used, if you have used this paper chromatography, you are using capillaries for spotting, and that are the capillaries, small fine capillaries will be used in which samples will be separated by keeping both the ends in the two of the electrode buffers. Okay, this is the principle. Now we are going in detail about this idea of capillary electrophoresis. So there is a concept here, that is theoretical plate number. What is theoretical plate number? Denoted by N. That is nothing but, sometime back in our discussion we were mentioning and discussing about the resolution, right? Resolution means the ability of the technique to resolve, and this ability of the technique will be better if you can resolve n number of components. Means, like we discussed, by SDS page, for example, 50 components you can separate it out, and by using two-dimensional gel electrophoresis, the same sample by SDS where you have defined 50 sample components in that sample, because of 2D gel electrophoresis, you are able to separate into 1000 components. That means that the technique is with a very high resolution, the ability to separate. Okay, understood what is the term or meaning for the resolution, right? So now this theoretical plate number refers to the ability of the technique to separate the components into n number of components. So higher the theoretical plates means the resolution of the technique will be high, and this resolution and theoretical plate number will be high when your voltage applied will be higher. But there is a limitation here. With the increase in voltage, your biological samples are vulnerable to get denatured. It will get denatured, and it will be even spoil. You can't run at a high voltage. So there is a need for controlling this heat dissipation. What happens with increase in voltage? There is increase in heat dissipation will be there, and that heat dissipation will be controlled by capillary electrophoresis. As heat dissipation is controlled, you can still run the samples at a very high voltage, and you are running at a very high voltage because you are able to control the heat dissipation effectively. That's how your resolution and theoretical plate number is very high in capillary electrophoresis. Understood? So this is the basic principle for capillary electrophoresis. Electrophoresis in capillaries. Separation of charged molecules under the electric field strength within the small capillaries, because the heat dissipation is very effective in capillary electrophoresis. Control of heat dissipation is very effective. So higher voltage is feasible. That's how you can achieve better resolution and higher theoretical plate number. This is the basis of capillary electrophoresis. And in the slide, there is an expression. N is theoretical plates, that is number of sample components is directly related to the voltage that has been applied. So higher the voltage, higher will be the number of component that you can separate it out, nothing but resolution. But you need to have the effective heat dissipation system to control this capillary electrophoresis. So by performing electrophoresis in the thin capillaries with small internal volume. So capillary now, it can hold your material, and capillaries are lengthy. They are like coiled and very lengthy in its structure. So you can fill, and at any point of any position in the capillaries, you can see there is small amount of the column matrix will be there within that. And what happens because of that, during electrophoresis, it is feasible for you to apply high voltage, because heat dissipation in such type of capillaries will be very effective, as it is holding minimum volume, but surface area you are increasing. This is the logic. Understood now? So you are making the matrix or material into small packed capillary tube, so that at any position you have smaller volume of the material. So whatever heat you can increase, easily you can maintain, right? So that way, these capillaries are very effective, and up to 5 to 50 kilovolts, 50,000 volts. But what about the normal or whatever we have discussed till now? That will be, you are using 250 volts, right? Have you used electrophoresis apparatus and this power pack? You know, if you recollect, like 100 volts, we are using 50 to 100 volts. Maximum SDS page 250, 200 volts we use. But in capillary electrophoresis, almost it is 10 times, 50,000 volts. Now it is enough for you to appreciate, right? How much voltage that you are applying, how much heat dissipation. If you had felt, if you had done the experiment and felt how much heat dissipation after running the SDS page, and you can imagine now 50,000 how much heat dissipation that you can expect. But in capillary electrophoresis, that heat dissipation is controlled very effectively. Okay, that is the principle. When you increase the heat dissipation, you are observing better resolution, better theoretical plate number. This is the concept of capillary electrophoresis. Any questions? All of you understood what I said? Okay, so no questions. I take it like granted that you all understood. There is another concept in capillary electrophoresis. So within the capillaries, these are silica coated walls are there, and here there is one phenomena taking place in capillary electrophoresis that is known as electroosmosis. In capillary electrophoresis, electroosmosis will be observed. What is this electroosmosis? So now this is the wall of your capillaries. These are coated with the silica, and there is a negatively charged, and over that a layer of counter ions, positive sodium and hydrogen. Above these ions, along with the solvation layer, that is hydration layer, water molecules, it will exhibit during the electrophoresis in capillary electrophoresis. It will move towards the cathode. Okay, so this all moves towards the cathode. This is the process observed. Now you see here, this is your electrodes, both the electrodes here, and these are the molecules present. One is electrophoretic flow. Electrophoretic flow, when you apply electric field strength, that is responsible for separating negative towards positive, positive molecules towards negative. That is electrophoresis, usual law of attraction. Now what happens, because of electroosmotic flow, opposite direction will be observed for negative. So these negative ions, anions, will move towards the cathode. Okay, so here what happens? You understood now? Earlier I told cations, anions, that is, there is no confusion. Chloride anions, sodium cations. Cations move towards cathode. So cation, sodium positive, cation moving towards cathode is opposite, that is negative charge. So this is cathode here. So this electrode opposing the usual electrophoresis, negative will be moving towards negative. Understood? That is the electroosmosis. So this electroosmosis is because of this counter ions and hydration layer that is moving towards cathode. This will takes these negative molecules also towards the negative electrode, that is cathode. This process phenomenon.
So there will be repulsion, no? So how is it possible?
Yes, yes, that is because of the hydration layer. Hydration layer, along with this hydration layer, there is a force which will overcome this electrophoretic flow that we are going to discuss. Okay, it's supposed to be repulsion, attraction between positive and negative, but here the opposite. So that attraction repulsion is followed in the electrophoretic flow, but electroosmotic flow refers to the opposite phenomenon, and that way your the negative ions will be moving towards the cathode. Okay, this is because of the electroosmotic flow. So capillaries with the negatively charged silica and layer of counter ions along with hydration layer are responsible for creating this type of force, and that affect only on the negative molecule, that is anions. Positive, there is no problem. Positive, anyway, it is moving towards the opposite electrode. So it will be plus effect, correct? Electrophoretic plus effect, and plus another plus effect is because of the electroosmotic. But this effect will be kind of a retardation effect for the anions, or the effect of electroosmosis can be a kind of a disturbance for the movement of these charged molecules in the capillary electrophoresis, and this is because of the electroosmosis. Understood? So now, as Amita has mentioned, that there is a opposite of attraction, though it is a repulsion, but it is being carried away along with the hydration layer, negative molecules towards negative. So what you have to do is that you need to maintain or reduce this electroosmosis. And how to reduce this electroosmosis in the capillary electrophoresis? That is by changing the pH. So you need to optimize the pH of the condition in such a way that electroosmosis is under the control and electrophoresis will be carried on. Okay. Then there is another phenomenon that is known as capillary isotachophoresis. So capillary isotachophoresis is a kind of application in which the capillary electrophoresis, during electrophoresis, the sample molecule will form or they will get rearranged in such a way that these ions will be with the different electrophoretic mobilities, such a way that there is the trailing and leading ions movement. In between that there will be sample ions, and this particular pattern of flow will be observed in isotachophoresis. So the rate of mobilities of leading and the terminating trailing ions will be forming a sandwich for the sample ions. These are the sample ions. So like this, as a stacking, it keeps moving during the capillary electrophoresis, and this phenomenon is called isotachophoresis. So this is the way. So you have stronger field strength and weaker field strength in between the samples getting distributed based on their characteristic electrophoretic mobilities, and this technique is called isotachophoresis. So this is the part of capillary electrophoresis. So we discussed in the capillary electrophoresis, the principle mainly relies on the high voltage and how effectively this high voltage can be controlled in capillary electrophoresis. Then we discussed about the way how capillary electrophoresis includes the phenomena of electroosmosis and then tachophoresis. So these two are the concepts of capillary electrophoresis. Moving on further, the equipment of capillary electrophoresis, this illustration as we have discussed earlier, these are the small capillaries, there is a detector, and this capillary, the two ends are connected to the buffer reservoirs, which acts as anode and cathode. And there is a sample zone through which you can introduce your sample. There is a detector which is connected to the data acquisition. There you can analyze. So this is a kind of window through which you can observe the sample movement. Okay. So at a high voltage, which is a characteristic of your capillary electrophoresis, about 5 to 50 kilovolts that we have discussed, but usually maximum 250 volts for usual electrophoretic technique that we use, but it can be increased to 50,000 kilovolts in capillary electrophoresis. Under that electric field strength, these capillaries are very effective for heat dissipation management system. If you see here, the heat generation at such a high voltage, it is about 125 degrees centigrade per mm. Per mm space, you can observe in capillary electrophoresis about 125 degrees centigrade. At that temperature, your proteins, everything get charred. So you can't do any technique. So because of the forced air cooling system, this temperature falls down to 51 degrees centigrade per mm. And further, there is a temperature controlled aluminum plate will be used in capillary electrophoresis, and this is responsible for further decreasing in the temperature to 7 degrees. So finally, about 125 degrees centigrade per mm temperature without any effective heat dissipation management system can be controlled to 7 degrees after using this heat dissipation control systems. So you have now basically 7 degrees, which is very good for your protein samples to separate. That is the uniqueness of capillary electrophoresis. Now these proteins, peptides, and nucleic acid, there is a laser-induced fluorescence, and that is the basis for detection, and through the window you can observe the sample, and it is connected automatically to the analyzer. And if you see, this is the point of interrogation, and the movement of these samples can be monitored, and this will appear in the detector as a peaks. So these each peaks are the components of your sample. So such a way, even at the 10 power minus 18 moles of precision, this is attomole. So you know picomole, right? This is 10 power minus 18 is attomole. At that particular sensitivity, you can resolve your samples and observe in capillary electrophoresis. So this capillary electrophoresis has got a main application in forensic science. Forensic science is known for the archived samples and old historic samples for investigation. So in such type of materials, even attomole sensitive components can be resolved by using capillary electrophoresis. In molecular biology, for DNA fragment separation, in PCR, DNA typing studies, and polymorphism, STR, these are all the different applications where capillary electrophoresis is also used. And to determine different ions like calcium, magnesium, sodium, potassium also, in saliva, this technique is used. And diagnostic purposes, capillary electrophoresis has got many applications, like in cancer biology, then hereditary diseases for its characterization. Even abnormal hemoglobin detection can also be analyzed by using capillary electrophoresis. So this is all for the capillary electrophoresis, the basic material that I have for you all with the information that I given. And any questions, discussion that now we can.
Sir, how capillary electrophoresis is used for detecting the tumor-related mutations, sir?
So mutations, basically single gene, it can be, right, suppose single point mutation, that is single nucleotide. Now, as we discussed, the resolution, separation is very precise, very, very sensitive. So even the change at one nucleotide can differentiate samples from that of the normal, and that is all how you can detect. So you have sample, that is your tumor sample with the particular mutations, and you have the control. Okay, and this is the technique with a very high specificity and resolution, and even the change at single nucleotide you can detect.
So is it based on the peak we can make out, sir? In the detector, peaks are formed.
Yes. So these are
based on this.
So
there will be changes in this peak.
You have to you have to have two graphs. One is control sample, and you have to compare control and sample. Test sample.
Okay.
So if there is any change, means that particular nucleotide or that particular position, there is a change.
Yes.
Analysis you have to learn from Biocon only, because they will be telling you, for depending on the application, you need to understand how it can be used. Any other questions, Himani?
Sir, what are tandem, sir? Tandem repeat. Short tandem repeats.
STRs, I didn't give in detail. These are biological concepts because chemistry students and all. So short tandem repeats, these are all different types of changes that you can expect in DNA. Okay. So here, as I mentioned, for example, sensitivity. You need to appreciate here, it is with an automole sensitivity, the changes between the normal wild type control samples and in the test sample. That can be precisely, this technique will pick up the changes. So you can easily pinpoint where the changes have taken place. So this is one of the type of change, short tandem repeat, that is taking place in the DNA, like mutation.
Okay.
Any other questions? Okay, further, if you have any questions, doubts, clarifications, you can mail through Girish sir, and I can answer to that. Otherwise, I'm done with today's session. Okay. So I wish you all the best for your future endeavors, and Happy New Year and good luck. Thank you.
Thank you, sir.
Thank you, sir.
Thank you, sir.
And Happy New Year to you too.
Happy New Year, sir.
Thank you, sir.
So much.
Thank you, sir.